about that time. So the time slot is from 4 to 5. Hopefully everyone's energy isn't waning. I know we're kind of getting to the afternoon here, and I just gave this talk 15 minutes prior, so hopefully I can make it through. So we didn't have a ton of time for Q&A after because there is so much material to get through. So just to preface this really quickly, I will be over here at the end. There is a quiz that you guys, I don't know if it's required or not, but there is a quiz at the end. So we'll end, I'll leave up the code for the quiz, and then I'll just kind of hang back if you want to talk. And then also we have a booth out kind of close to the main stage by where the food is. And again, happy to continue the discussion there. So first of all, before we get into it, just by a show of hands, because the last group, I think it was about the same size, I asked, how many of you have taken a microbiome test? And I only got 2 hands. Okay, be honest, because I think there's a lot of liars in that last group. How many people have taken a microbiome test before? Okay, we got 2, 3, 4. So just 4 people? Okay, well maybe they were right. Okay, so how many people have heard of the microbiome before? Okay, a lot more. So we're going to specifically be looking at the gut microbiome. And hopefully by the end of this, you'll see the importance of actually profiling this ecosystem, and it is an ecosystem, and potential utility for what you're doing. And at the very least, having a better understanding of what exactly the microbiome is, how it ties to gut health, but overall systemic health, because it does affect many different organ systems within our body, all the way from the liver to the brain. So just to kind of make sure we're set on definitions. So when I refer to microbiomics, I'm just referring to profiling the microbiome. The microbiome is referring to the actual genes that we're profiling, whether we're looking at the skin microbiome, oral microbiome, vaginal microbiome, or more commonly, the gut microbiome. So usually we'll perform metagenomics to do that. Another term you might hear is called the gut microbiota. That's just referring to the individual microorganisms, not their genetics. So just to make that distinction, and I find myself very often guilty of interchanging those, and I can't guarantee I won't do it during the talk, but just so you know. So the discussion or the talk that I'm going to give today is looking at how to optimize gut health, particularly focusing on profiling the microbiome and how the microbiome can actually be leveraged through a clinical lens, whether this be through modulation of that environment or specific recommendations, whether they be dietary, supplemental, lifestyle. We'll talk about some very brief pharmacological applications as well. So specifically, the outline, we're going to be going through some very basics about the gut microbiome. Some of this, of course, you may know. I'm going to be going a little bit deeper in some areas, so hopefully I don't lose you. I think the questions aren't too crazy, too in-depth, so I'm not going to help you, though, so don't ask for me to help you with cheating. We'll look at some strategies for gut health, and we'll kind of see where the future of this space is going, particularly with the way to construct research in this area, moving away from things like randomized controlled trials and getting more into N-of-1 type trials. Learning objectives, I'm not going to rehash this. I think we've kind of already talked about this. I don't like to talk about myself, but very briefly, I received my PhD at Arizona State University back in 2022, specifically in the area of nutritional microbiology. I actually still am in academia. I have 1 foot in academia, 1 foot out in industry, so I'm currently a postdoctoral fellow under a T32, which is essentially a training grant under the NIH, and my focus is in artificial intelligence for precision nutrition. Again, my specialty is microbiomics as well as multi-omic integration. We will discuss what multi-omics means later on. And I am the co-founder and director of microbiomics at Therium. Again, we have a booth out by the main stage. Obviously, I'm employed by Therium, right? Okay, understanding the gut microbiome. So in each and every one of us, unless you've had some resectioning of your gastrointestinal tract, we have the alimentary canal extending from your mouth out the other side to your anus. we do collect a lot of great sample for profiling the gut microbiome. However, we can use other technologies. Biopsy is one if we're wanting to look at the small intestines, for instance. And there's also lab-on-chip technologies that are now under development where you can actually swallow a pill, and this pill can actually take subsamples at different regions of your gastrointestinal tract. And then obviously you have to defecate it, and then we can take that and profile different spatial omics of the microbiome. So there's a lot of exciting things going on in this space, but I always like to kind of start from first principles, so we'll start off with a little A&P. This canal is about 20 to 30 feet. Obviously, when we first learn about the gastrointestinal tract, it's all about digestion, absorption, and assimilation of nutrients. All these things are occurring in different regions. However, in parallel to that, we have these different environmental constraints, whether we're looking at oxygen levels, whether we're looking at pH, bile acids, short-chain fatty acids. Obviously, transit time is going to affect these things with the actual biomass of principally bacteria, but we also have other kingdoms of microorganisms. So the deeper you go down into the gastrointestinal tract, you're going to have more dense populations of microorganisms, principally in the colon, right? So we have about 90 to 95% of overall abundance of the microbiome, the gut microbiome, is bacteria. We also have other kingdoms, less abundance, obviously, like the archaeome. We have the mycobiome, so this is actually comprising things like fungi, as well as protists and even viruses. Okay, so now we have this whole new field coming into the fore with the virome, looking particularly at therapies like bacteriophages, if you have heard about them. So we'll talk about all those different things as we go through the talk. We also know that in terms of Western societies, there's a high prevalence of not only gastrointestinal symptomology, something that actually can be implemented if you own a clinic quite easily, and if you're focused on gastrointestinal health, is things like the Bristol Stool Scale. So this is a validated rough proxy for transit time, essentially, and we're going to get some, things we're going to talk about poop today, unfortunately. So just bear with me. So the Bristol Stool Scale is this essentially a Likert scale with poop images. So you can have, essentially like little rabbit hard poops, which show slower transit time all the way up to things like diarrhea. It's actually not a bad proxy to use. It doesn't really cost you anything. There's also the gastrointestinal symptomology rating scale. This is a 15 questionnaire. It's based on a Likert scale from 1 to 7, where you can really quickly establish gastrointestinal symptomology, upper and lower, so you can look at things like heartburn, you can look at constipation, diarrhea, et cetera. That's actually a really great tracking tool, and again, something that can be easily implemented in your practice. And actually, we use to inform a lot of our analyses in conjunction with what we're doing through molecular biology. But a lot of those symptomologies and profiles that we're detecting in the microbiome are tied to a lot of these prevalent diseases, whether we're looking at things like irritable bowel disease, whether we're looking at SIBO, whether we're looking at Crohn's, ulcerative colitis, celiac disease, et cetera. The prevalence has really risen over the years, so this has really kind of brought profiling the microbiome under the spotlight and also having utility in other areas, as we'll see. Now, a lot of these actually directly relate to our immune system and also meta-inflammation. About 70% of the body's immune system is located within or around this structure, most commonly termed the gastrointestinal associated lymphatic tissue, which you may have heard about. So in conjunction with profiling this community, we often also want to look at inflammatory cytokines, different immune signals. These are going to be really important conjoining barometers that we can use. Okay, again, I do like to go kind of take first principles approach and kind of take also a historical perspective. And I do apologize if the resolution is not good. I don't know if people in the back can even read this, so you'll have to listen to my monotone voice, unfortunately. Back in 1677, Antoine von Leeuwenhoek was actually able to use a pretty rudimentary form of microscopy where he's taking these different lenses and looking at different media, whether it be lake water, they looked at snot basically, spit, rainwater. He was seeing these little moving organisms, which he actually called animalcules. He didn't call them the microbiome, but he described these as life forms. So these were microscopic, meaning that they are not visible to the human eye. So this is where we get the term the microbiome, microscopic. Now fast forward to 1842. Antoine von Leeuwenhoek did not profile these in the gut. That was actually Dr. John Goodsir, who looked at gastrointestinal fluid, and again, through microscopy, identified moving organisms, these microorganisms. Dr. Ilya Mekhanov, back in 1907, he's really the one that came up with the idea that these microorganisms in our gut are intricately tied to our health. He made observations of rural populations in Europe consuming a lot of fermented foods, fermented yogurts, fermented vegetables like pickles and sauerkraut. They were living very robust lives, not having a lot of allergies, and not having a lot of common colds and things of that nature. So he hypothesized that beneficial microbes have a beneficial effect on our health. And actually, we named our microbiome test at Theriome after Dr. Mekhanov because of how important and how fundamental he was to this field. Because then we start really see things picking up. Now, a lot of these observations were constrained by the technologies of the past. Obviously, we talked about microscopy. Culturing methods were starting to be used more and more. In 1977, it was found that certain types of Bacteroides, bacteria could actually ferment different polysaccharides, particularly fibers, and actually produce different important components in our gut, like mucus. Now, the real growth was seen on the tailcoats of the Human Genome Project. So, of course, everyone knows, really spearheaded by Craig Ventnor, this enabled a powerful sequencing technology to profile what was going on in the genome. And this could be applied not only to humans, but obviously other larger organisms, other vertebrates like mice, rats, but also profiling microorganisms. Back in the early 2000s, this technology was really quite widely used, particularly with pyrosequencing and 16S sequencing, which we will discuss what exactly that means. But that really caused a boom in this field and all the computational and bioinformatic tools that were necessary to handle this big data. Because now we're not looking at one genome, we're looking at potentially thousands of different genomes within the gut environment. And that's again why we call it metagenomics when we're profiling things like the gut, because we're looking at the overall collection of genomes that we're profiling. We started to tie form to function, looking at a lot of animal work in this space, actually finding that dietary composition affected the types of microorganisms that we found in the gut. So diet was early on seen as a very important modulator of the overall ecosystem. Also, actually metabolizing different food substrates and creating beneficial metabolites that could then enter into circulation. So the gut was then seen as this essentially a mediating variable connecting what we're eating to ultimately what we're seeing in circulation in our body, and then obviously going and getting deposited in various different organs, and even exercise. So we all know that when you exercise, you can accumulate lactate. And it was thought a while ago that lactate was just kind of a waste product. We now know you can actually perform gluconeogenesis in the liver through the Cori cycle and actually recycle it. However, we also have a recycling mechanism in our gut microbiome. This lactate can actually enter into the luminal space and interact with specific microorganisms that can actually create beneficial short-chain fatty acids, particularly propionine. So, how do we acquire the microbiome? Well, most of the seeding actually occurs right after birth. There is evidence that actually some of it actually does occur in the womb, but not to the degree that with colonization events at birth. That's why vaginal births are actually very important. In fact, it wasn't often done when C-sections were performed years ago that we would actually perform an inoculation of like a vaginal swab on the child. We now do that because we've recognized that this is one of the primary moments where we have that that transfer from the mother to infant. So this develops throughout infancy, even still formalizing into puberty. Not only is this important for creating that ecosystem, but also all the important interactions that are going on with actually maturing the immune system, because again, we know that there's so many immune interactions occurring with the galt tissue. Once we're into adulthood, the microbiome actually becomes quite stable, and for a healthy individual, quite resilient, meaning that it's more difficult to actually shift that state. Now, diseased individuals can show varying levels of resilience. That's an important metric to profile because usually resilience is seen as a beneficial health metric. However, if someone is in a dysbiotic state, say they have Crohn's disease or IBS, and they are resilient, that means that we're going to have to find those anchoring elements that are holding them there and essentially depopulate that or get rid of those or remove some of those functions that are holding them in that state to shift them out of that dysbiotic configuration. So for the microbiome and modulating the microbiome, it's not as easy as going to Costco and taking a probiotic or the kitchen sink. Really, particularly when you're in adulthood, you want to do a profiling to see what metrics you need to actually address if you need to shift that state. Again, this is quite layman. I think it's a decent analogy. When we talk about the microbiome, again, we're trying to get away from this reductionist approach where we're really hyper-focused on one specific type of bacteria or microorganism. That doesn't mean that that doesn't have clinical utility, particularly if we're looking at, say, C. diff or something of that nature. However, when we're doing a global profile, we want to pull in tenets from ecology, particularly community diversity and community structure. So usually we want to have increased diversity, and simply what we're referring to here is the number of species or strains that your microbiome is harboring. So you can think of this as the analogy with the rainforest. In a healthy rainforest, right, we're going to have a lot of ecological diversity. We're going to have different types of plants, we're going to have different types of animals, et cetera. And the reason that this is beneficial is if we have some type of perturbation, we're going to be able to recover a lot faster because we have other organisms around that to kind of pick up the slack if we get a reduction. So, you know, with the hamburger bulldozers and the slash and burn French fry diet, a lot of people might have. This is not a good configuration, obviously, because if we have further assaults, whether they be antibiotic or whatever, it's going to be a lot more difficult for that person's microbiome to recover and for beneficial microorganisms to recolonize. In fact, you know, that's one of the issues that we get with people that have had their appendix removed. The appendix was considered kind of this inert add-on or remnant organ. We actually house a lot of biofilm in the appendix that kind of re-inoculates our overall gut microbiome when we do have a pretty bad perturbation like heavy dose antibiotics, et cetera. So with someone that has that structure removed, usually after a heavy course of antibiotics, this is something that we do want to profile and make sure that we're encouraging repopulation of the appropriate core microbiome. Now, very interesting about the gut microbiome, in terms of actual functional potential, it exceeds what we have in our own genome, okay? There's different estimates of how many genes you have in your microbiome versus your host tissue. I've seen this number reduced over the years. Essentially, it's a little bit over 1 right now, according to the most recent estimates. However, in terms of overall biomass, if I was actually able to go into your alimentary canal and extract all those microbes, remove digesta, remove fluid, it's about 200 grams, so something that you can fit in the palm or your cupped hands essentially. So it's really not very much biomass, but it has tremendous functional potential, affecting a host of different factors, whether we're looking at harvesting food energy. We'll get into short chain fatty acids and bile acid modulation later on, improving overall gut function, Obviously, gut barrier. I don't like to use the term leaky gut, but most people understand what we're talking about when we use that term. Obviously, producing a suite of potentially healthy and potentially detrimental metabolites. In fact, about anywhere from 1/3 to 1/2 of metabolites in circulation have been modified or actually produced by the microbiome, which you can really see that this is a massive influence on our overall health. Obviously, boosting the immune system, also synthesizing a suite of different vitamins and hormones. We know, for instance, a lot of B vitamins are produced by these microbes, vitamin K, as well as different hormones actually related to appetite regulation. So there's actually mechanisms not dissimilar to GLP-1 hormones. Obviously, now we have GLP-1 agonists, so you can just take those instead, I guess. But there are things that we can do to affect things like appetite and even communication with satiety with the connection to our brain. So as I mentioned, principally when we look at the gastrointestinal tract as a structure and in terms of its function, digestion and absorption. So in the small intestine, that's what it says right there if you can't read it. Here's the brush border. Here's food particles coming in, right? A lot of that is occurring in the small intestine. So we'll digest carbohydrates, proteins, obviously breaking them down to amino acids, fats, but a lot of different substrate digested does come into the colon and then obviously into contact with these higher density biomass areas, particularly fiber. And the reason that fiber is seen as so important in terms of microbiome health, and you can kind of fit prebiotics under that definition, is the production of 3 principal short-chain fatty acids. Normally in a targeted panel, we're looking at about 14, but the most abundant would be acetate, first, butyrate second, and then propionate. And these all have different effects on the body because the small size of these and their volatility, they can actually get absorbed by the gut epithelia quite readily and enter into circulation. For instance, acetate has a beneficial effect on muscle tissue as well as the brain. Butyrate actually acts quite locally as well as other structures like fat tissue. The great thing about having good levels of butyrate is the epithelium, these brush border cells, their principal substrate is actually butyrate. So if we have good amounts of butyrate, we're fueling them so they can be healthy and they can carry out their functions, particularly related to absorbing food, but also maintaining adhesion to each other through tight junction proteins. So we can have, we don't have those issues with gut permeability, which can be quite problematic, particularly related to things like endotoxins. So endotoxins, really, when we profile endotoxins, we're looking at one in particular, and there's many different subtypes. They're called lipopolysaccharides, LPS. So when we have turnover of microorganisms, particularly gram-negative bacteria, we know that these microorganisms aren't living very long, right? There's constant die-off, they lyse. These LPS structures on the outside of those cell walls, if we don't have robust levels of mucus coming from goblet cells and surrounding this space between the epithelium and the interluminal contents, that's one thing, but if we don't have a good regulation of gut permeability, these can pass quite easily into circulation, interact with the liver, and spur meta-inflammation. We know meta-inflammation is tied to most chronic diseases that we see now in the modern world. So very interesting with short-chain fatty acids, butyrate that I was just talking about, it can actually get absorbed, as I said, into circulation and interact with white adipose tissue and actually have a beiging effect, meaning that we can increase mitochondrial density in these fat tissue deposits and make it more akin to brown adipose tissue. So we know about brown adipose tissue, right? It's more thermogenically active, meaning that we're going to be burning more fatty acids. It's more metabolically healthy to have increased levels of brown adipose tissue in our body. So it can actually directly help with things like fat loss, as well as improving your overall metabolic profile. Acetate can actually cross the blood-brain barrier and have satiety effects. So again, we'll focus a little bit more locally for this talk because we're focused on the gut, but just understand that there are a lot of different axes that are present throughout the body interacting with the microbiome. One really important one that you'll probably hear more about is bile acids. We know from the liver we produce bile, and the gallbladder gets secreted to help digestion of fat. Some of those do enter into the colon, where they can be deconjugated, particularly by microorganisms that express high levels of BHS, bile salt hydrogenase. Okay, so particularly species like lactobacillus, for instance, and bifidobacterium express a lot of these. The reason that can be a good thing is we produce these secondary bile acids that can actually act as signaling agents locally at the gut barrier, and particularly related to two mechanisms. One would be XFR, related to lipid metabolism, or glucose metabolism, and then TGF-5 related to glucose metabolism. So we can actually positively modulate circulating levels of sugars in our body as well as our lipid profiles. So as I said, there's many different axes. The gut-brain axis, I think probably most people in here have probably heard of this. It's just really fascinating to, from a mechanistic standpoint, identify these different metabolites that are coming from the gut and affecting these what we would consider faraway structures, right, from the gut microbiota. So there's a lot of implications here, particularly for profiling and addressing different health states. And we can look at everything from the gut-brain axis, the gut-liver axis, the adipose tissue. We can look locally, as well as overall meta-inflammation, again, which can be spurred from things like LPS, endotoxin. Okay, so let's shift gears a little bit. We'll look at common strategies used to assess just gastrointestinal health, as well as the gut microbiome. So a lot of these have been around for a long period of time. You're probably familiar with many of these. Clinically, a lot of these are still very valid. It certainly doesn't, I'm not trying to dismiss these at all. I'm just saying that, for profiling this environment, I think bridging these and integrating a lot of these metrics with the microbiome is really going to be the way forward. We have obviously more traditional tests like fecal occult blood tests. We can do immunochemistry. We can do stool cultures, which I know actually this is quite popular now for particularly looking at pathogens and parasites. Obviously, we have profiling the microbiome. There's even more distal proxies, like we can do breath tests if we're looking at like breath hydrogen, things of that nature. There's also we can do blood tests looking at different immune markers. Those are still valid, fine. And also some really, some cool stuff that I talked about with the lab-on-chip technology, where we can swallow capsules and collect different regions in the gastrointestinal tract. And then, of course, metabolomics, which is looking at metabolite expression. And we can look at this in the feces, we can look at this in the blood, we can really look at this in any tissue. So I'm not going to go too deeply here. I'm just going to draw some distinctions and get you a little attuned to the vocabulary that's used in this space, particularly the sequencing space. So as I mentioned, in the early 2000s, we had sequencing technology really come into the fore and enable us to do this type of profiling. Really the workhorse, and in many cases it still is the workhorse, particularly for clinical research, is what we call 16S rRNA sequencing, ribosomal RNA. So we're actually not looking at DNA when we perform this type of analysis. We're looking at RNA. RNA, as you can see here at the structure, when we profile rodent and human microbiomes, we're usually interested in these 9 variable regions, okay? So usually for 16S with human work, you'll see the V4 region be kind of considered the gold standard. We're basically looking at variable regions on that RNA structure, which we can then bank against a library and then annotate what type of microbe that is. Now, as I said, there's 9 hypervariable regions. There is new technology available where we can profile all 9, and we can get some resolution to the species level. So to step back for a second, tree of life, We might start from the kingdom where we're talking about bacteria, virome, archaeome, et cetera. We might go down, getting a little bit more granular, might be looking at the family level, genus level, species. So usually on a probiotic bottle, for instance, if you look at the back, you'll see the species listed. Sometimes they'll list strain. They really should be listing the strain. If they aren't, they're not a good company. And then we have at the strain level. So taxonomy is good, but one of the limitations here is we're only really able to, with confidence, if we're just looking at, say, like the V4 region, get down to the genus level. Now, long read, where we're pulling in all 9 of those hypervariable regions, we can get to the species, but we're still kind of missing some things. We're missing strain, and we're missing functionality. So we can have add-ons to this as well. Again, 16S is usually done for bacteria. If we're wanting to look at something like eukaryotes or fungi, we're having to do some extra profiling here. So it can get not cost effective, usually if you're using a company that's profiling 16S really constricted to bacteria, which is missing out on the whole story. So enter in metagenomics, okay? So this is now being considered the gold standard. Really, if you are going to profile the microbiome, particularly the gut microbiome, you want to use this approach. You should make sure that your vendor, if you're doing this type of analysis, is using, whole metagenomics. So what we do is through some pretty sophisticated chemistry processes, we lyse the cells, the bacteria or the other microorganisms down, we get at their nucleus, we extract out the DNA, and we essentially break it apart into these tiny little fragments, hence the term shotgun. So this is called shotgun metagenomics. Based on our sequencing capacity, we obviously can't profile these long strands. We need to break them down into more manageable chunks. We then sequence those fragments. It's like a puzzle piece. We then recompile them into these long contigs, and then we can actually infer, with our base pairs, what they are taxonomically, what they are in terms of taxonomy, and then also what they're doing. So function is extremely important. Understanding what's there is good. You really want to know what the heck they're doing. So this is really where metagenomics shines. There are also other options within metagenomics. You can do shallow sequencing. Usually, this is what a lot of companies are doing with 2 to 3 million base pairs. But to get down at those deeper functional levels, you're wanting to do more deeper sequencing. Usually, 10 million base pairs is kind of considered good. Obviously, you can go more. So just a quick side-by-side. With 16S, obviously, we're looking at the ribosome. With shotgun sequencing, we're looking at DNA. It's going to be more expensive because there is a lot more computation involved in the extra extraction processes. However, I think the trade-off is justified. We're getting better resolution at the taxonomic and species level, particularly getting down to the strain. We're detecting these other kingdoms that we are understanding are very important in terms of modulating the overall microbiome, particularly the virome. And also, we have really validated tools for making sure we eliminate contamination. So when we do shotgun metagenomics, we actually have to control or eliminate for host tissue, because if you think about enterocytes, these cells that are lining our gastrointestinal tract, they're dying every 2 to 3 days. They're sloughing off. They're actually coming out into stool. So we have to actually align those sequences with the human genome to remove that noise, because that's obviously going to interrupt our analysis. Same thing with different food components, chloroplasts, things of that nature. We want to remove all that background noise to make sure we have a very representative data to work with to profile strictly the microbiome. Now, there are obviously other omes. There's so many different iterations of omes now, but the core group is genomics, transcriptomics, proteomics, and metabolomics. Now, when we talk about the microbiome, we're obviously looking at multiple organisms. So that's where you'll see meta in front of each one of those, right? So we've talked about metagenomics. There's also metatranscriptomics, metaproteomics, and essentially metabolomics, right? Looking at the full suite of metabolites in a sample. Other ones like lipidomics, it's kind of like a subsection of metabolomics, but these are really the core group that you want to know and kind of focus on. All right, Shifting gears again, we're going to be looking at strategies for improving gastrointestinal health. So here's a really important point, and I think a misunderstanding might be an artifact of a lot of the marketing material with things like probiotics. Your microbiome is as unique as your fingerprint. Not everyone has the same microbiome, right? So you're really colonization limited, meaning that not everyone has the same profile, in varying abundances, just waiting for, that microbe waiting for the right opportunity to grow in abundance. It may not be present at all, and we may have a real challenge actually even getting that microorganism to grow in that environment. A good case in point is Akkermansia muciniphila. If you've heard about this, they've actually commercialized it. It's now available as a probiotic. However, there is a percentage of the population that does not have this microorganism. The reason that this microorganism is deemed so important for health is it resides in the mucus that surrounds the brush border, right? And as I said before, we don't want those luminal contents to come into contact really in the colon directly with the brush border. We want this buffer zone. That's what the mucus offers. Akkermansia you can kind of consider as a lawnmower in the mucus. It does degrade mucus, but also encourages the growth of mucus because you don't want an overproduction of mucus. You want a good level, and you want to encourage goblet cells to be producing those proteins, those glycoproteins. So Akkermansia, usually in a normal population, we'll see anywhere from, to put a really rough figure, 1 to 5% of total abundance. But some individuals do not have this. So does that mean that we should be taking all these different types of probiotics if it's not going to graft? Probably not. We might want to look at other strategies. So that's where profiling, if you're looking at the microbiome as a lever to change, you're really going to want to profile it versus taking something indiscriminately. Now, there are opportunities throughout the lifespan to pull that lever. This is work that we did at Arizona State University. I'm not going to go too deeply into these graphics, first of all, because the resolution is terrible, and I'm kind of guessing everyone's educational level here in terms of the microbiome. Hopefully, we're all still with each other. But essentially, we looked at the microbiome over the academic year of college students in a dormitory. So we controlled the types of foods they were eating. They were actually fairly transparent about the amount of alcohol they were drinking, surprisingly. So these individuals are actually considered adolescents. They were 18 to 19, so kind of in those later stages. Microbiome is still forming, not obviously to a lesser degree what you'd get in infancy. But what we found when we actually looked at the overall structure, and we had over 500 individuals in this cohort, so a fairly large sample, we saw this branching of different configurations. We were principally interested in some of the most dominant microorganisms that were kind of seen as orchestrators of the overall community. And what we found was there was 3 big groupings, and then we had some variability. So the first one was Ruminococcus, which was a lesser degree. We also had Prevotella, which is usually associated with a more fiber-rich plant-based diet, and then we had a clustering with Bacteroides. As we track these individuals over time, looking at metabolites, so what was in the feces and also in the blood, we found that those individuals that had a Bacteroides dominant configuration, so again, more associated with a dysbiotic configuration, they were producing a lot of problematic metabolites. We saw cholesterol sulfate, for instance. We saw increases in p-cresol sulfate, which is actually tied to degradation of kidneys. So we started to see some early signs of essentially a poor health trajectory. Now, when this was identified, we were able to try to course correct many of these individuals, which was not successful in some cases. But in many cases, we were able to get them on a better track and then actually see lower expression of these problematic metabolites. So the microbiome is malleable, but you kind of have to understand baseline composition before you go in and start modulating that environment. Now, in adulthood, because we have this stability, we have to look at pervasive and chronic pressures. So we're all eating for the most part on a daily basis. So diet is actually one of the main modulators. Unfortunately, in the West, not only are we usually not consuming enough fiber, but we're also getting a lot of different assaults, whether we're looking at food emulsifiers, for instance. So in this particular study, this is a rat study. Again, you probably can't read this. So our control group was consuming water. We had another group consuming carboxymethyl cellulose, which is a food emulsifier, and then polysorbate 80, which is a food dye. We found, not surprisingly, that in the animals that were consuming those food additives, they had an increase in fat mass, dysregulated metabolism, but really importantly, when we looked at the mucosa in the gut, this is mucus right here, and these are the brush border cells. In comparison to the control, which had a nice healthy layer of mucosa, we saw degradation occurring in the animals fed those food emulsifiers, and also suppression of overall microbial biomass. So, while the FDA may deem these as generally recognized as safe GRAS status, we seem to see that this doesn't have a very good effect on your microbiome. Now, in comparison, we know there's very simple things that we can do, particularly consuming more fiber, more fermentable food, and quite rapidly in a compromised microbiome, we can see increases in diversity over time, as well as quelling of pro-inflammatory cytokines by implementing these fairly easy dietary strategies. Another really pervasive problem, in fact, over 50% of people that have IBS actually have also small intestinal bacterial overgrowth, SIBO. Okay, so this is a very common problem that I'm sure you're all. Essentially, we have encroachment of contents in the colon, microbial contents in the colon start to come up into the small intestines. We don't really want this, particularly a lot of the fungal microorganisms doing this, because not only is it going to interrupt our digestive process and absorption, but in terms of what the patient's feeling, it's also related to a lot of symptoms, right? So there's a lot of treatments available. I know the first go-to for many is antibiotics. However, we know that, again, there's a lot of different levers to pull. There's dietary modification. Low FODMAP diet we know can be very effective. Probiotic supplementation, again, targeted. Prebiotics, so you're really thinking, not to be confused with probiotic, a prebiotic is really providing food to beneficial microorganisms so they can thrive and populate. There's also herbal remedies, digestive enzymes, prokinetics, stress management, and I think often neglected intermittent fasting. So this is actually an area of work that we've delved into over the last few years. And what we know, when someone does implement an effective intermittent fasting or time-restricted feeding strategy, we're actually able to give what we term gut rest, okay? So for most in the West, particularly in places like America or the Americas, we're usually in our waking state in a postprandial state, meaning that we're constantly digesting food. If we're eating breakfast, lunch, and dinner, and we're constantly snacking, we're never giving time for the gut to rest. The mechanism is fairly well known. It's essentially a motor complex in our gut that's, think of it like peristalsis, actually moving contents along. You have to really be in a fasted state for this to work effectively. It does kind of a reset every 90 to 20 minutes. So even doing some fairly simple time-restricted fasting, or time-restricted feeding, and intermittent fasting can actually be a very effective way to encourage that and move things along, because we do want healthy transit time. We don't want things sitting, particularly in the case of SIBO, because then that's going to give opportunity for those microorganisms to encroach upon those areas. So we got to discuss probiotics. Obviously, we're talking about gut health and the microbiome. I do have to touch on it. I will point you to the ISPP's official definition of a probiotic. In my opinion, it's extremely vague. Probiotics are comprised of live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. That could be a lot of things, right? Now, the interesting thing is probiotics, according to one of the more recent large-scale surveys that I looked at, the United States is the third most popular dietary supplement. So a lot of people are taking these. I'm not sure they know why exactly. They may have a vague idea of why they're taking them, but they don't really know if they're actually working. Now, there's many different formats. Obviously, we have just straight supplements in capsule form. Obviously, yogurts have them, fermented foods, kefir, kombucha, cheeses even, and we have them now. Manufacturers have done some interesting thing with shots and drinks, and there's even gummies, available. So there's a lot of different formats. But I just want to, again, kind of take a step back. If we don't really have a good understanding of what type of environment we're introducing these microbes, think about this. If we were to take out the alimentary canal, the gastrointestinal tract, and cut it open and map it out so we could actually get a calculation of surface area, we're talking about 200 square meters. So for us in the US, that translates to over 2,000 square feet, on par with a single family home, right? Now you're talking with a probiotic, how many colony forming units, if the company even lists them, are you taking? Maybe 5, 10, 15, 50, 100? It's really a literal drop in the bucket in terms of this massively complex environment. And we know a lot of these interactions, as I said prior, are occurring through the immune system. So it really doesn't make sense to take these products indiscriminately. It really makes sense to understand what is going to be the best product that's going to work with your environment. In terms of profiling the body of literature that's out there, we performed a pretty comprehensive systematic review in healthy individuals looking at the effects of probiotics in randomized control trials. For the most part, they really didn't have much of an effect. And again, I don't think probiotics don't work. I just don't think they're being used correctly. In terms of ecological pressures, kind of giving a hierarchy, and this is not the rule of thumb. This is just, based on the body of literature, what we're finding in adulthood to really change the microbiome. If we need to do that, principally it's going to be diet, okay? And when we're forming our reports, a lot of clients don't like to hear this. They want the silver bullet. I understand that. I understand easy solutions. That doesn't mean there's not a place and time for supplementation or working with their medical practitioner, maybe some pharmacological interventions. But we know, again, just based on the constants and the pervasiveness of diet, it's really going to be one of the main things that you want to focus on for a healthy microbiome. And all the other things that we kind of, in many cases, no. Obviously, we can dose these things more precise with individuals, but it's not till we get to these lower portions that we see that in terms of the body of evidence, probiotics are going to have generally a less of an effect size. Again, not the rule of thumb. So this kind of brings us to personalized gut health. So going back to the ohms, right? I'll bring up multi-omics, man. He's kind of washed out. Again, I think this is just the projector, but essentially when we look at the human, in terms of what we term the systems biology framework, okay, that's a term that you'll probably hear more in the future. This is kind of the paradigm or the dogma of biology. We start with the genome, right? So this is the code of life. Now, just stagnantly sitting in the nucleus of the cell, it's not really doing us very much good. So we need these messengers to take that code and come out into the cell and actually do something with it, right? So those is what we term the transcriptome. Obviously, we want some kind of functionality. That's going to be our proteins, our proteome. So here we're making things like enzymes, we're making structures, we're making hormones, things that are actually doing something in the cell, going out into our body and doing something. Downstream from that, we get the expression of metabolites, okay? So this is what you can think of as metabolites, or the metabolome, pardon me. So here we're looking at all the different substrates going into different fuel cycles, various different amino acids, basically all the metabolites that we're detecting in any particular sample. And then we have the microbiome, which has its own systems biology paradigm. We've kind of just been talking about genomics, but it also has a transcriptome, a proteome, and then obviously the fecal metabolome. Now, the reason that we really like to focus on the microbiome and also the metabolome is based on their longitudinal expression. Unless you're doing some kind of nefarious things with like gene editing and stuff, you're really not going to be shifting your genome very much, right? You're not really taking this multiple times. Transcriptome, it can shift, but not to the degree that you would get with global metabolism or the microbiome, and the proteome is pretty stagnant. So in terms of shifting metabolism or shifting that ecosystem, really the focus that we've taken is looking at the metabolome and the microbiome. And this is actually what we did in this paper published in 2024. We actually did do a randomized control trial, but also a responder analysis that we'll get into, really kind of calling for a more personalized profiling, a more personalized approach when we do these types of studies. So really briefly, what we did is a parallel group design. We randomized individuals either to an intermittent fasting type diet or a standard calorie-restricted diet following a TLC, therapeutic lifestyle changes. This is what the Academy of Nutrition and Dietetics advocates, particularly for obese individuals with cardiometabolic derangement. So these individuals were matched for energy and generally macronutrients. Now, over the course of this 8-week study, and again, it's very hard to see, but the individuals following the standard calorie-restricted TLC diet did lose weight, but not to the degree that the intermittent fasting individuals did. They lost significantly more weight. However, we saw variation, quite substantial variation in this response. This is looking at relative weight loss, okay, not absolute weight loss. So bank that for a second. Now, in terms of the microbiome, again, talking about that disruptive event that intermittent fasting beneficially can cause in the microbiome, notably, we saw an increase in diversity in these individuals, as well as overall community structure. Again, I'm not going to describe in detail what these metrics are. I'm happy to stand off to the side and field questions after. Now, what was driving that was when we performed this differential abundance analysis, at the family level. So normally in dietary studies of the microbiome, we might see some shifts at the species level. We might see going up one at the genus level. It's very rare to see things happen at the family level. We saw stark differences in abundance of microbes at the family level in the group that followed an intermittent fasting diet. Many of these associated with a lean phenotype, particularly Christinolaceae. You see here was our strongest hit. Rickenolaceae, Ruminococcaceae, increased, and these have actually been tied to lipolytic activity and effectively digesting proteins, producing beneficial amino acids. So here we saw a really positive shift in these obese individuals following an intermittent fasting diet. And this was also reflected in their inflammatory panels, particularly with anti-inflammatory cytokines. However, going back to that variability that we noticed in weight loss, we saw some individuals, have less of an overall or relative weight loss. That might be fine for some individuals, but then we saw these really hyper responders. So we were interested in looking at difference in profiles pre and post between these low responders and these high responders. And what we found was in the low responders, a more resilient microbiome, more attuned to actually extracting energy out of the food that they were consuming. So here they were very resistant to change, and we're trying to essentially hold on to body weight. Whereas the individuals that had a very high success rate, lost a lot more weight, showed a more flexible microbiome and the ability to really metabolize different types of lipids, activate different amino acid pathways, and really produce a more healthy profile. So, when approaching these types of randomized control trials, I think, first of all, a lot of these really should be crossover design, but really, we want to take a more N of one approach because we know, again, everyone's microbiome is extremely individual, and not everything is going to work, in a group type setting. So we were able to track an individual and actually perform some digital twinning, which I'll discuss in a moment. So we tracked an individual. Data I'm displaying is over here. We actually tracked this individual for 2 years. He was compliant, actually over 95% compliant on his regimen because he was very motivated to lose weight and to get healthier. But we tracked him monthly, and what we found with appropriately assessing his metabolic profile and his microbiome, we're able to be agile with our recommendations, make proper course corrections, and see that reflected in his microbiomic profile. So again, you can't really see these species very well, but this is essentially looking at different populations of microorganisms over time in that individual. And we saw blooms of very health-associated microbes, particularly Akkermansia, mucinophilia, increased phenomenally in this individual. And this was also in conjunction with improved metabolic profile and weight loss. So we see many different levers that we can pull, whether they be compositional, if we're focusing on, from a macronutrient, a micronutrient level. Obviously, there's different feeding cycles that we can implement. The microbiome is a very important mediator with the foods that we're taking in, how we're taking in those foods, and then us, the host. So in terms of this complex environment, we really think that if you are going to target it, again, you should be profiling. And this is really amenable to artificial intelligence work that we've been doing for a couple of years now, particularly in something called digital twinning. So originally in aerospace, they had this problem of testing out different parts under different simulations. It was very expensive to put those parts through real world conditions. So they created computer simulations to put stresses on those structures to effectively save money, right? We've adopted that engineering approach now in systems biology, looking at metabolism if we're looking at the metabolite, or now the microbiome to perform these simulations if we're looking at a library of different recommendations, what we know in the literature to effectively move different populations or affect different functions, and see what is going to work best for that person. So we can have an in silico version of that person, and if they were compliant with the recommendations, we could expect to see changes in that profile versus in reality, right? I mean, obviously, we can't fix the issue of compliance, and that's something that I'm sure a lot of you guys deal with. But we're moving towards, I think, a model that is really going to get at the heart of precision types approaches. So, to summarize, I think you guys have a decent understanding of what the microbiome is at this point. Hopefully, we all kind of understood the different sequencing technologies that are out there and why deeper metagenomic sequencing is really kind of the way to go. And I think, according to Mohr's law, and we've seen this with sequencing in general, the cost is going to decrease over time. We really want to take personalized approaches. There's different technologies emerging. Digital twinning is just one. That's an approach that we like. But I think, the approaches with big data, there's going to be a lot of other solutions coming out. And ultimately, we can apply this beyond the gut microbiome, whether we're even looking at things that we have done profiled traditionally over time, like CMPs and other types of blood work that we can feed into these types of very advanced models. So with that, I will end. Again, my company is Theriome. We are located in downtown Phoenix. As I said, I will be by the booth. If you want to stop by, I can give you my contact information if you're ever in town or if you are local. We are happy to give lab tours and talk with you in greater depth. And I really appreciate your time, and I wish you the best of luck on your quiz.